Hello. Thank you again for joining us for this presentation, The Evolution of Benchtop NMR Spectrometers, Technology and Applications from 60 to 100 Megahertz. It was originally given as a CNE News webinar on June 10th, and we're happy to make it on demand here for you again. My name is Susanne Riegel. I'm the Director of Marketing and NMR Product Manager at Nanalysis. I've been here for about 10 years, helping to define and launch a series of our Benchtop NMR spectrometers. This talk will be about 40 minutes, so please stay with us to the end, and please be sure to reach out if you have any additional questions. Benchtop NMR spectrometers have been on the market for about a decade now, so people are becoming increasingly familiar with them. However, I would like to start by putting them into context a little bit. I've compiled a brief, extremely brief, summary of NMR spectroscopy from the discovery of important properties of atomic nuclei in the early 1920s, namely electron spin and magnetic moments, to the development of the first continuous wave NMR spectrometers that were made from surplus radar supplies post-war. This rapidly facilitated the discovery of things like chemical shift and coupling constants that brought us through into the 1950s. With all of these properties, it was quite clear that commercially NMR spectroscopy could provide a great deal of value, and surely the first commercial NMR spectrometers were made available and permanent magnet-based 60 megahertz instruments. So these were rapidly joined on the market by superconducting magnets that immediately started moving to higher and higher fields. In the 2000s, this was as high as 1,000 megahertz or 1 gigahertz, and then of course in 2019, Bruker announced their 1.2 gigahertz platform. The idea being, as you move to higher field, you improve the performance, sort of the basic physics that underlying the NMR experiment. At higher fields, you see higher sensitivity and higher resolution. If we look at the research that paralleled or drove these instrumentation advances over time, you'll see that the ones that received the highest recognition strongly focused on things like pulse programming and application development, where you could extract more and more information from increasingly complex samples. Perhaps the most notably of those would have been Ernst with his 2D experiments, and regrettably, he just passed away. Given these trends, it came as a surprise to many when Nanalysis announced the launch of their 60 megahertz instrument and PicoSpin their 43 megahertz instrument in 2012. I remember many early discussions in the field amongst thought leaders about whether these instruments would provide value and help further NMR, or what their potential might be. Basically, the question lingered: why would you take an instrument that has largely focused on improving performance and return it back to low field? Well, the answer, of course, is accessibility and affordability. If we look at the geographical distribution of NMR spectroscopy, you can see about 67% of the world's instrumentation is located in the United States and Canada and Europe, and there's very limited access in other locations throughout the world, as reported by the SDI Global Assessment Report in 2019. If we look at this even more granularly, you can see that there are 8 countries that have 80% of the world's spectrometers, with the United States, Germany, China, Japan, and the UK rounding out the top 5 countries. NMR is also strongly stratified by application. You'll see another chart here from SDI, where they estimate that 76% of NMR usage is for qualitative structure identification or elucidation type applications in academia, government, pharma, and biotech. Contract research organizations make up a great deal of the NMR community as well. But you can see in industry, we only see strong representation in agricultural and food sciences, and the adoption in others has been comparatively quite low compared to research-based organizations. So we view accessibility as one of the main limitations for NMR spectroscopy, and one of the main limitations that Benchtop NMR can start to address. If you think about magnetic resonance technology in sort of a continuum, people who are in synthetic chemical research are very familiar with high-field superconducting NMR spectrometers, where people who are in specific industries probably picture the low-field, low-resolution relaxometers, which perform bulk property measurements for standardized testing, such as solid fat content, spin particle coating, etc. So the concept behind the Benchtop NMR would be to just add another layer of NMR workflow. They were not at all meant to replace high-field NMR spectrometers, especially when it comes to increasingly complex molecules that have limited solubility or isolated in small quantities, like perhaps a natural product. But they could offer more information than strictly time domain data about the sample. So the Benchtop NMR has a wide potential range of applications with increased resolution and sensitivity relative to a relaxometer, and the ability to perform structure identification and elucidation tasks, do basic reaction monitoring, etc. But small enough and affordable enough that it can be productized for a specific industrial application, whether or not you have an NMR expert present in the field. So throughout the course of this talk, I'm going to talk a little bit about Nanalysis' technology that we've used to make our 60 and 100 megahertz products lines. I will frame the benchtop low-field NMR in terms of workflow and spectral quality to explain how we view the benchtop NMR position in the market, whether that be for research, teaching, or test and measurement spaces. We will then move on to discuss the applications of benchtop NMR for underserved high-field users, namely chemical education, startup, chemical, and SME. We will then discuss the vision for new NMR markets through method development, productization, and application validation. So the Nanalysis Benchtop NMR product line is based on neodymium iron boron magnets configured in compact, patented, and highly homogeneous Hallback-based arrays. The Hallback is important for two main reasons. One, they have very strong internal magnetic fields, but little to no stray field. So the instruments are safe to sight and to be around, and then the second reason is that they have a symmetric opening in the top and the bottom of the instrument that helps to accommodate flow applications, where you can think of the NMR as an online detector for whatever you're trying to monitor. So because our magnetic fields are based on permanent magnets, all you need to power them is a standard electrical outlet. There's little to no maintenance because the spectrometers are cryogen-free and do not require weekly or monthly nitrogen or helium fills. The electronics have been miniaturized onto small circuit boards, stacks, and they are included within the same enclosure as the magnet to ensure that the instruments are portable and easy to sight. While the instrument can easily be controlled from an external computer that's either directly connected to the instrument or networked in remotely, it is not required to have an external computer. These are built-in touchscreens that allow you to do onboard data processing and acquisition. We feel that this was necessary to help ensure that the instruments can be placed anywhere. In early 2020, Nanalysis acquired RS2D, a high-end NMR electronics company located just outside of Strasbourg, France, based on their high-end FPGA-based magnetic resonance spectrometer technology, or what's better known as their Chameleon 4 platform. RS2D uses this platform to power their line of magnetic resonance technologies, ranging from MRI consoles to replacement high-field NMR consoles known as the Pulse, as well as their DNP module called the Gecko. We identified this platform as an excellent addition to our 100 megahertz instrument to accelerate the collection of high-quality data and fast-track the release of new features like gradient pulse-based programs. Nanalysis has two permanent magnet-based products, the 60 megahertz 1.4 Tesla platform and the 100 megahertz 2.35 Tesla platform. Both come in proton-only or multi-nuclear configurations, proton, carbon, fluorine, and phosphorus perhaps being the most common, but there are other nuclei available as well. Each instrument is equipped with standard 1D and 2D experiments, depending on which probe was purchased with the instrument. Using the CAM4 technology, we are able to perform more experiments on the 100, and we're also able to offer an advanced pulse programming module through the RS2D software called SPINIT. So for those that have done a fair bit of pulse programming, you know that typically it requires that one learn a new pulse programming language. One of the nice things about SPINIT is that the SPINIT does not require the use of any code. The user is able to just define the sequence of elements in the graphical user interface, and the software automatically converts this graphic into an appropriate code. The user can interact directly by defining pulses, delays, phases, etc. in a very simple and user-friendly way. So for example here, you can define your pulse and then hit solve, and the program automatically determines what that means for the hardware to ensure that your pulse is applied with the specified amplitude, width angle, and type. To understand the relationship of external field strength and spectral data, I thought I would show some ibuprofen spectra. So not a particularly interesting molecule, but it's often used as a benchmark molecule to look at the resolution in Benchtop NMR. So I predicted this from 60 to 700 megahertz and shown it in the standard ppm x-axis. So this helps to illustrate the closely related terms of resolution, dispersion, and spectral complexity. These terms are very closely related, but they are not fully synonymous. Resolution refers to whether or not two peaks are separated from each other. But the first thing to notice is that at 60 megahertz, the peaks appear shorter and fatter. And then as we start to move to the higher frequencies, you'll notice that just visually, the peaks begin to look taller and narrower. So they have better resolution as you move to higher field. But what we're actually observing is a dispersion issue. So it's particularly noticeable in the aromatic region at around 7 ppm, where you can see that what's clearly an AB quartet at 60, 80, and 100 megahertz starts to turn into a doublet of doublets at 400 and 700 megahertz. It can also be seen in the quartet at about 3.75, which becomes increasingly sharp. And then finally, and perhaps the most noticeably, in the isopropyl methine septet that's at around 1.9 ppm. So this overlaps with its neighbors quite substantially at 60 megahertz, and you cannot make out that full septet. It comes a little bit clearer at 80, but you can see the whole septet, but it's not fully resolved from the next peak. And then as you move to 100, you can see that that methine finally becomes a distinctive peak. And then of course, as you move to 400 and 700, that distance continues even further. So in ibuprofen, we see better resolution at higher field, but this is not so much due to a second order effect as we might expect, but rather to dispersion. So just to refresh everybody's memory, we typically see a second order effect when the chemical shift in hertz between signals, say A and B, is not greater than or equal to 5 times the coupling constant between those. So it's very common to see second order effects in aromatic regions and areas where the coupling constants are quite small. But this isn't really the factor in ibuprofen that affects the resolution. Instead, it's primarily a dispersion related issue. So to help illustrate this, I again have used that same predicted ibuprofen spectrum, but instead of using the normalized ppm x-axis like we're used to seeing, I've instead displayed the x-axis in hertz. So the dispersion at 60 megahertz is there's 60 hertz per ppm. So in a standard 10 ppm spectral width, you only have about a 600 hertz distance over which the resonance will be displayed. At 100 megahertz, there's 100 hertz per ppm, therefore 1,000 hertz spectral width. Whereas if you go all the way up to 700, this is a 7,000 hertz window. So seemingly what you see is that the spectrum gets stretched out and all of those resonances are much clearer and much further separated. So at higher field, there's both less chance to observe second order effects, and we often see less spectral overlap. This can also be illustrated with the centrosymmetric ortho-substituted benzene rings, such as diethyl phthalate, which was acquired on 60 and 100. So we would expect to see a complex and characteristic pattern of what we know to be an AA prime, B, B prime spin system in the Popel notation because of the line symmetry. So we have 2 distinct aromatic resonances, A and B, with this line of symmetry, but they are magnetically inequivalent. So instead of seeing one signal for each, we see this AA prime and then B prime characterization. So in the early days of continuous wave spectrometers, ortho-substituted benzene rings were often used to determine and assess the resolution of a particular instrument. And although that's not the case today with Fourier transform-based instruments, they have a very distinctive characteristic line width. So at 60 megahertz, you can see that the A prime, B prime signals are running into each other, where they are fully separate at 100, and we do see a distinct A and B resonance. So now that we're done discussing the factors to be considered when looking at different field strengths, I would like to start talking about how benchtop NMR are being used currently and how they can be used in the future. So the main first adopters of benchtop NMR is what I would call the underserved users. So these are groups of people that don't have sufficient access to high field NMR spectrometers, and so they would like to be able to supplement the access that they have. So the distinctive groups of people in this category would be those in chemical education, whether they're teaching organic, inorganic, analytical, or biochemistry, as well as startup and small and medium-sized chemical companies. But additionally, there is interest in the development of benchtop NMR for quantitative applications, whether that be for reaction monitoring or process analytical technology, or the development of very specific QA/QC screener or analyzers that can provide very specific assays for those who need to determine a value of something. So to just highlight the chemical education use case, the value of incorporating hands-on use of benchtop NMR spectrometers is immediately apparent to any chemical educator. To help students learn meaningfully, it is well known that they need to have a good cognitive understanding of the material, they need to have generated effective feelings about that material, and especially in the case of an experimental science like chemistry, they need to develop the necessary psychomotor skills. However, despite being one of the most commonly reported characterization techniques in literature, the average undergraduate student does not get hands-on access to a spectrometer. So benchtop NMR can provide tremendous value. The accessibility is such that you can easily incorporate into an undergraduate lab. But the problem that it potentially introduces is the one that I've just described, and that's the problem of resolution. So, however, resolution issues are often associated specifically with low field NMR. I just wanted to point out that resolution is a problem in all forms of measurement, and it's not specifically unique to NMR spectroscopy. So there are ways to optimize it and to get around it. So one of the things that you can do when trying to manage spectral complexity is you could rely on heteronuclear spectroscopy instead of proton NMR. Typically, this means that you're moving to a wider spectral width and you have less resonances with which have the potential to overlap. So something like phosphorus or fluorine are very commonly used as NMR handles to see what's happening in your spectra without having to worry about complexity or spectral overlap. Instead of relying primarily on 1D NMR spectroscopy, which is often done, you can introduce multiple pulse experiments to extract correlations or perform pure shift experiments. Software tools like peak fitting, quantum mechanical modeling could be used, or even the development of complex molecules using chemiometrics or machine learning techniques can also help to extract more information, regardless of whether or not they're visually well resolved. Hyphenated methods can help to separate mixtures and make spectra a little bit cleaner. But what's unique to academic teaching is that you also have the unique opportunity to strategically select molecules that don't show higher field coupling. So if you're interested in this, there's an extensive list in this paper in the Journal of Chemical Education. So there's a number of different examples that you could pick, but for the purpose of this presentation, I have selected indomethacine. So of course, this can be synthesized in a very simple reaction whereby the starting material and the product have very unique chemical shifts when they close to form this indole ring. Despite the predominantly aromatic signals at 100, you can see in the spectrum, I can clearly make out individual resonances for the AB quartet in the para-substituted benzyl chloride moiety of this molecule, and then 3 unique doublets in the indole portion of that. If I zoom into the aromatic region here at 60 megahertz, you can see that you can only sort of make out a multiplet at 60. So 100 really helps to further the structural elucidation of this molecule. Another example that we have on our website is actually looking at the purity of indomethacine using a malic acid-based internal calibrant, and this can be done at any field strength as the resolution is sufficient. The second underserved user that I would like to highlight is those who are performing chemical synthesis. So there has been growth worldwide in the number of micro, small, and medium-sized chemical companies. 96% of chemical companies in Europe, for example, are considered to fall into the SME category. And in the US, SMEs are increasing the number of exports and contributing 144% to the number of which they export. So to better understand this demographic, we have paired with Spirochem, a 10-year-old contract research organization based in Basel, Switzerland, that specializes in the pharma and biotech space. So while we've sort of developed our understanding of this market vertical with the CRO, pharma, and biotech lens, it really is important to note that while there are many sectors performing chemical synthesis, the molecules that they're making are dramatically different, but the synthetic workflow that they follow often remains quite similar. So this can range from agrochemicals to materials and polymers to flavor and fragrance, for example. And in this talk, I will focus primarily on pharmaceuticals, agrochemicals, and food science. So typically, chemical synthesis workflow involves setting up your experiment, monitoring the reaction, working it up, purifying those products, analyzing those products, and drawing your conclusions. So if the reaction is deemed successful, one can just move on. But however, we all know that usually this requires some optimization to improve yield or reduce byproducts. So typically, you repeat the cycle over and over until you're happy with the final synthetic procedure. So if you had, it was the perfect world, you would typically include NMR in various stages. So that's each that I have with the starred magnifying glass there. So that would be during the monitoring of the reaction, the purification, as well as the analyzing of that reaction. So to incorporate NMR spectroscopy, an SME is usually faced with a choice to either purchase a high-field NMR. So typically this would be an initial cost of about $250,000 and an annual about approximately $110,000 a year for maintenance fee, which would include both the cryogens to upkeep the instrument as well as the person to maintain that instrument. But you get your data quite quickly and it would be high-quality data. You could also choose to outsource to an NMR facility. So depending on the location and the availability of the NMR facility, typically standard service is about $50 per proton NMR and $250 to $500 for carbon NMR, but unfortunately it takes about a week back to get your data. So if you needed to accelerate this, you can of course usually opt to pay for priority service, but typically while you get your data faster, the cost adds up quite considerably here, and you're not incentivized to use NMR more, you're incentivized to use NMR less. So depending on the number of chemists per site, the company's revenue, typically you have to be a certain point before you're ready to purchase your high-field NMR spectrometer. So many people are forced to rely on outsourcing NMR, and they don't incorporate NMR with the frequency with which they desire. So this is really where Benchtop NMR can supplement the workflow to save time and money in outsourcing costs while still increasing the amount of data that's marked at each step throughout the course of the reaction. So the 100 megahertz Benchtop NMR is perhaps most useful for this demographic. To help illustrate this, I would like to highlight two agrochemicals, the first being diphenate, or an organothiophosphate-based insecticide. At 60 megahertz, this has a very distinctive and interesting coupling pattern, but it is highly second order, and the chemical shifts and coupling constants cannot be extracted by the human eye. So I've sort of just put a example report here. You would see 4 basic multiplets over a wide range of the chemical shift area. At 100 megahertz, on the other hand, we still see some multiplets. We can extract all the chemical shifts and coupling information, but in the alkyl region, for example, we can start to observe the traditional triplet pattern that we would understand visually and start to get some chemical shift information and therefore structure elucidation of this molecule. By combining standard 1D and 2D NMR experiments, you can start to elucidate the structure, even in the circumstance that the spectrum is not fully resolved, or you're observing second-order effects that prevent the full visual assignment of each resonance. So we can run a COSY of diphenate to help identify the ethyl fragments and differentiate the ethyl and ethoxy groups, even though the methyl triplates are partially overlapping. So starting with the multiplet at 1.6 to 2.2 ppm, we see 2 off-diagonal correlations with the triplets centered at 1.1 ppm and 1.34 ppm, suggesting that this is not, in fact, 2 triplets in the 1D, but instead we're seeing one triplet and a doublet of triplets that overlaps with the first triplet. So this suggests that we're seeing heteronuclear coupling for the methyl protons and the phosphorus group. The multiplet at 3.8 to 4.6 ppm, which integrates to 2, is the methylene of the ethoxy fragment, and it correlates only to the triplet at 1.35, confirming that there is no observed 4-bond proton-phosphorus coupling through the oxygen in this fragment. The assignment from the COSY can be further confirmed with a heteronuclear 2D multiplicity edited HSQC. By adding multiplicity editing to this experiment, we can help to differentiate between the CH and CH3 groups, which will appear in one phase, and the CH2 components, which will appear in the other phase, very similar to the output of the perhaps better known DEPT 135 experiment. The first correlation we see is between the large and small triplet resonances, confirming our assignment of the methyl group as a doublet of triplets. The second correlation lines up again with the large triplet, confirming once again that it is in fact a triplet, not a doublet of triplets. These first two signals are both positive in their phase, as we would expect for the methyl group. The next two multiplets are blue because they're in the opposite phase of the red methyl groups, confirming again that they are the CH2 components of the ethyl fragments. The first of these two correlates to the large carbon doublet, which is due to the direct 1-bond phosphorus-carbon coupling of about 83 Hertz, and the second to a much smaller but still observable 2-bond coupling of about 7 Hertz, which can be assigned as the methylene of the ethoxy chemical shift. In the aromatic region, we see two distinctive correlations of the meta-para and carbons, which correlate to the multiplet in the proton NMR spectrum. So, while using the 100 1D and 2D data, we are able to assign all the peaks in the proton and carbon spectrum, but we're still not able to extract all the relevant chemical shift and coupling constant information that we require. But what's interesting about the 100 megahertz is that we do have enough resolution to start using added software layers such as quantum mechanical modeling, and I'm going to highlight here using NMR solutions to do so. So to analyze the experiment data, NMR Solutions requires that the structure and the starting value for the chemical shift and the coupling constant be input into the software. The software then iterates on these to match a theoretical result with the observed spectrum until a metric gets a suitable fit. So the metric is this pentagon 5-point parameter here that matches things like intensity, chemical shift similarity, and coupling constant similarity. The output shows the experimental in blue, the theoretical in red, and the difference in green. So you can see how well the theoretical and the experimental data directly match. But from this, it is possible to observe high-level coupling and realize all the chemical shifts and coupling constants so that we don't have to use the report of a multiplet at any point throughout the spectrum. So you can see that they're quite complicated here. So we're showing doublets of doublets of doublets of doublets of doublets, or doublets of doublets of quartets, but we're actually able to get all of the chemical shift information in this complex molecule. So the power of this software is discussed if you're interested in learning more. I would highly recommend this Guido Pauli paper, where they use it to understand the primary, secondary, and tertiary polypeptide structures using high field and low field NMR data. To highlight with one final example, I would like to use chloranthroniloprol, a rhinoid insecticide. At 60 megahertz, the aromatic region is largely dominated by multiplets, whereas at 100 megahertz, we can start to see the expected doublets come out and behave in a far more first-order manner. As you can clearly see here, a nice doublet of doublets. If we use NMR Solutions, we can, of course, extract all the structural information and remove any report of multiplets from that. And you can see using my pentagon fit metric here, we have an excellent fit between the experimental and the actual data. To expand the use of Benchtop NMR spectrometers, my application chemistry team has focused specifically on method development. So by this, I mean that we specifically explore potential analyses of using Benchtop NMR and what would be required in terms of pulse programs, signal processing, etc., to be able to use the Benchtop NMR to quantify specific values at an accurate, precise, and simple way. Once we've identified where there's a value proposition for using NMR, I work with our product development team to define the market requirements, whether that be through performance specifications, increased ruggedization, sample method modification, or increased connectivity. This allows us to validate a method and provide a market-driven solution. So we've done the majority of our method development at 60 megahertz, and I wanted to highlight 3 case studies using the 60 megahertz NMR. The first being a very traditional use of NMR spectroscopy, basically unknown structure identification. So designer drugs, and by that I mean those that have very small structural modifications such that they maintain the desired psychological effect, but the structure modification means that it can't be identified using traditional targeted methods. So Benchtop NMR could be very highly useful in this regard because it's untargeted and we can use it to directly determine the structure. We're not matching it to a known standard. So to be able to put an NMR spectrometer directly into a lab, of course, it needs to be compact and it needs to be portable. But for it to be reliable enough that it could provide useful data that would potentially hold up in court or give forensic scientists confidence, we need accurate and reliable data. We need the instrument to be extremely stable, both to temperatures and vibrations, whether that be small vibrations due to driving of the instrument or hitting perhaps a large pothole. The spectrometer has to be still, and it has to consume a small amount of power such that it can run off a backup generator. And one of the most important requirements of all is that it has an automated interface that allows data acquisition to be very simple, and it matches to a database reliably such that forensic scientists don't need to be NMR experts. So we're still working on the development of this, and we've initially paired with both ACD Labs and the LKA in Hanover. So we've studied a wide array of drugs, and we can identify both the active ingredient and filling agent in a series of street drugs compared to pure samples. For a second example, we've partnered with Sartec, a value-added partner that has developed a machine learning-based method to help us identify the properties of crude oil. So refineries that have a number of different types of incoming crude that have to be refined, each one of these incoming crudes has to be tested extensively to ensure that the refinery process is optimized and that the blends can mix throughout the process. So typically this requires offline methods that require 7 to 14 days. So they add up quite a lot to be able to hold the oil at the site and wait for the data to return. So using a patented method that they developed on Highfield, Sartec was able to extrapolate all of these experiments down to a basic 1D NMR. And so they used this patented method and extrapolated down from the 400 megahertz all the way down to 60 megahertz. So we could potentially introduce a crude oil screener at line at the refinery. So to simplify this for refinery technicians, using the Nanalysis application programmatic interface, Sartec was able to write an easy software program to ensure that Spectra was acquired and matched to the database, and those results were held with good data integrity such that they could be audited later. So this method is highlighted in this paper for anyone who would like to learn more about this application. The final example that I would like to highlight is the lithium content analyzer. Lithium is becoming increasingly important in modern society, as I think we all know, but methods for optimizing and analyzing lithium extraction fluids are typically based on atomic absorption or inductively coupled plasma, so they require extensive sample preparations, and they often suffer from strong matrix effects for other cations like magnesium or calcium that are present in the lithium broths. So by using lithium-7 NMR spectroscopy directly, you don't have to worry about basic matrix effects because, of course, things like magnesium and calcium will be silent in the lithium-7 channel. And additionally, we can use the brine fluid directly, and it requires no additional sample preparation. So we've highlighted this recently in a talk and in this paper here. So if you're interested, please access this paper, which was recently made open source by SQM, our collaborators on this application. So typically when exploring methods, we try to define the minimum viable tool required. And for the previous examples that I've shown, 60 megahertz usually provides enough resolution to be able to reliably extract your data. But there are some potential benefits from 100 megahertz in a series of applications, and the one I would like to highlight for this talk is in analyzing fraudulent food. So I think we're all aware food fraud is becoming increasingly problematic in today's society, and this just refers to when consumers purposely misled by either substitution, dilution, or addition of another substance other than the one that is being marketed. So while this is usually done exclusively for economic gain of the seller and is not usually harmful to the customers, this is of course not always the case. So two notable examples would be the toxic oil syndrome in Spain in 1981, where waste industrial oil was added to the olive oil and made a number of people sick, and a number of people unfortunately died as well. And in 2008, the Chinese milk scandal, where protein content was exaggerated in testing by the addition of melamine to milk, which required a lot of kidney problems in the infants that were drinking this milk. So for the purpose of this work, I would like to focus on using the 100 megahertz to screen olive oil and honey. So we first started exploring the efficacy of Benchtop NMR for the identification and quantification of soybean adulteration in extra virgin olive oil in 2014. So these figures are taken from that time, so I do apologize. They are not the highest quality compared to the other figures that you've seen. But as you can see from this profile taken here, well, all of the edible oils have the same characteristic regions for things like olefins or the glyceride linker or the terminal methyl groups. They do differ quite a lot here in this bisallylic region. So the bisallylic region is due to the unique CH2s that are located between two olefinic regions, as you can see here in the omega-3 and the omega-6. So olive oil is thought to be have only a small amount of omega-3 and 6, so this peak is quite small, whereas soybean oil is composed largely of linoleic acid or omega-6, and so you can see that this peak does quite dominate in that region. So using this resonance, it's quite easy to identify and quantify the addition of lower cost soybean oil to a more expensive olive oil. If we compare an extra virgin olive oil acquired at 60 megahertz with the same sample acquired at 100 megahertz, you can see a pretty marked difference in the resolution between those two spectra. So while the olefin and bisallylics are still well resolved at 100, you can see that the CH2 alphas to the esters separate from those, alpha to the olefins, and you can also see a distinctive resonance for the CH2 beta to the ester linkages. Upon examination of a wider group of edible oils, you can see a small peak resolve at about 1.0 ppm. Well, it does overlap with terminal methyls. This is important because instead of just relying on that bisallylic region to differentiate the different oils, we can actually start to quantify the fatty acid composition using this peak. So it's unique from the other peaks and allows us to do a full fatty acid profile. And this is directly indicative of the linoleic or the omega-3 content. In a comparison with extra virgin olive oil and canola oil taken at 100 megahertz, you can see that fitting the linoleic peak to determine its integration helps us to determine the amount of linoleic, or 18:3, and from the amount of linolenic, 18:2, or the amount of oleic, 18:1. From a quick analysis, we can see from our experimental values that the 100 match up quite nicely with the theoretical values, and we're continuing to explore the accuracy with which we can profile these fatty acids relative to high-field NMR instrumentation. For our final case study, I will explore a preliminary analysis of honey. Honey is one of the most adulterated foods because of its interest for its nutritional value and its enzymatic content. But it does have production limitations based on both the nectar of plants as well as the bee population. In Canada, we export about 60% of the 90 million pounds of honey that are produced here. While the adulteration of honey is not very well understood, it is thought to happen by potentially three methods. The first of which is direct adulteration, where non-honey sugar-based products are added directly to the honey. Secondly, it can be indirect, where the bees are fed a sugar product to increase their production value. And then finally, it can be the mixing of a lower cost honey with something that's a little bit higher cost. Unfortunately, testing methods for any of these have not been particularly effective. For plants classified as C4, isotopic analysis can help identify adulteration, but as the majority of plants are C3, there is some issues with this, and NMR is thought to potentially provide a lot of value here. To better understand the sugar profile, we will start by looking at sucrose, a disaccharide, and glucose, a 6-membered monosaccharide, and fructose, a 5-member monosaccharide. As you can see, there's a high degree of spectral complexity in the 100 megahertz data due to the similarity of the components within these rings. At 4.8, you can see a water peak in all three. Sucrose has a nice NMR handle with the proton next to the oxygen linker, and glucose for the anomeric proton next to the oxygen in the ring. If we look more closely at glucose, we can actually see two doublets. The one at 5.35 that's well resolved, and the one at 4.77 that's sort of straddling the water resonance. We know that glucose interconverts from its alpha to beta form through a mutarotation process. These distinctive forms can be identified where the anomeric proton is either in the axial or equatorial position, as depicted here. The beta form is more thermodynamically stable because of steric considerations of these two neighboring hydroxyl substituents. So if we look at the J-res, we can confirm our hypothesis through the chemical shifts, and you can clearly see the difference between this axial or equatorial form. And then if we use Messer Lab's QGSD feature to extrude these chemicals, we can integrate them, and you can actually see that they match pretty well with the theoretical ratio of 32 to 68 that we would expect to see at 25 degrees. So the 100 megahertz NMR spectrum of some purchased honeys and sugars show a pretty standard profile. Honey is made primarily of glucose, and we may see some adulteration in other samples that we have purchased, like the Al-Shifa or the pure honey. The C4-based plants are cane sugar, which is primarily sucrose and corn syrup, which is an observable mix of these two things. The C3-based plants, beet and maple, are primarily sources of sucrose. Rice syrup is a mixture of maltotriose, maltose, and glucose, effectively just glucose in the body, and you can again see the very distinctive resonances of all of these forms. So while this work is very preliminary, we are refining our sample procedure and acquiring data for a wider range of honeys to start building a training set to be able to identify the purity, potentially the origin, and of course the authenticity of these honey samples. So to conclude, I hope that I've showed you that Benchtop NMR provides an accessible, affordable, and automatable layer of magnetic resonance technology to further the use of NMR spectroscopy in both traditional NMR markets as well as into the search of new ones. Like high field NMR, Benchtop NMR has followed a trajectory moving from lower to higher field to help expand the number of applications and the number of samples with which it provides useful data. And finally, we believe that the future of compact NMR lies in focused responsiveness of technological innovations, method development that meet the needs of basic high return on investment type applications. So I'd like to thank the people that did this work, Matt for his studies on the olive oil, Paul for his work on honey, Thais for her spectral analysis, and the rest of the Nanalysis application chemistry and marketing teams. We'd like to thank Mike Bernstein at the Amber Analytical Services for his expertise in QNMR as well as software, Thomas Fassard for building out our SME market understanding and helping us to build the basic case study there, Matthias and Petrus for their help with quantum mechanical modeling and the development of the NMR solution software, the folks at the LKA for building the illicit drug database and helping us to do basic identification there, Demetrius at ACD Labs for the automation and population of that database, Sartek for their expertise in machine learning and crude oils, and then SQM for building the lithium analysis case study. And finally, you for your attention. Thank you very much for hanging in there. We'd be happy to take any questions via email, and please watch some of our other videos, including the 100 megahertz demo video. Thank you very much for your attention.